Because we've all got tablets, I want to be able to involve you in actually mutating, analyzing, interpreting how mutations affect protein coding genes. So that's kind of the exercise for today. It all starts with this particular gene sequence. I went onto an internet database and downloaded this CDS, which stands for coding DNA sequence, of a gene called nuo1. I just chose one at random. So here you see the DNA sequence of the coding DNA sequence. This is everything that was transcribed into messenger RNA minus, because this has already been spliced, this does not contain any intron sequence. So this is showing you a messenger RNA sequence, except it's used T's instead of U's. Hopefully that doesn't freak anybody out. It's a messenger RNA sequence, essentially. And what's colored with the background shading are the different exons. So yellow here is exon 1, and then we've got exon 2 next to it in a different color, exon 3, exon 4, exon 5. So this gene has 6 exons. What are the letters that don't have color behind them at the start and at the end? There's 4 nucleotides at the start of this sequence that are in lowercase that also at the same time don't have a background color. What does that mean? Say that again? What parts of messenger RNAs are not part of an exon? This happens to be what's called the untranslated region. So at this end of the messenger RNA is the 5' untranslated region, or UTR, untranslated region. Down here at the bottom, which I can't draw on, is the 3' untranslated region, the 3' end of the transcript. What do you think it means that they're untranslated? That's what it sounds like. Where's the start codon here? Be a eukaryotic ribosome. Where's the start codon? We're about to translate this messenger RNA. What is the start codon? What 3 nucleotides? No? Okay, go back to the codon table and tell me what the start codon is. Methionine. AUG, or ATG in this case. So where do we see the first ATG show up? So it's the first, right, it's the first set of capital letters. So there's 4 nucleotides, and then there's an ATG or AUG. So what it means to be a 5' untranslated region is that's part of the messenger RNA molecule. It's just that the ribosome reads across that part of the transcript until it finds the first AUG, the start codon. Hence, those 3 nucleotides are not translated. The ribosome doesn't read them and turn them into amino acids. It's an untranslated region. What should that tell us about the 3' untranslated region down here at the bottom? The white background, lowercase letters. What's just before the 3' untranslated region begins? TGA, what's that? That's the stop codon, right? So all of the colored, the background color tells us, hey look, the first part is the start codon, methionine, and the last codon is the stop codon, asterisk. All of the messenger RNA transcript that's past the stop codon doesn't get translated, it's untranslated. Ribosome stops translating at the stop codon. So all we really care about is the sequence that's highlighted, the exons. That's the part between the start codon and the stop codon that the ribosome is going to translate. So if I was really mean, although it's Friday, so I'm feeling pretty nice today, I'd ask you to translate all of this and tell me what's the amino acid sequence of this protein. But, we've got tablets, so don't do it by hand. Here's a useful website to use to do translations of messenger RNA sequences for you instead of using the codon table. So if you want to know the amino acid encoded by that CDS, coding DNA sequence, you go to this URL. There are lots of these web tools that are available. This is just one I happen to pick. It's not the best one necessarily. You go to this website, and right here, you paste in, oh my goodness, I'm using the lightsaber pointer. You paste in the CDS sequence there. The force is strong with me. Yeah, damn straight. And then all you have to do is down here at the bottom, just pick submit. So you paste in the CDS sequence, and then this computer program, the website, of course, just has the codon table built into it. It reads the CDS in groups of 3 nucleotides, and then tells you what they translate to. And so I paste in. I'm just walking you through this because I'm going to ask you over the weekend to do this yourself for your gene. So you'll have this recorded. It'll be on the YouTube movie, but I wanted to show you in class in case anybody had any questions. It's just this easy. Take a CDS, a gene sequence, paste it in, hit submit, and then in a slide, I'll show you what it looks like when we're done. So that's the program TranSeq. It's a website. You can paste in a gene sequence, and it tells you what protein sequence a ribosome would produce. What I want to know, one of the things I want to know from you over the weekend as you work on this exercise is how many amino acids long is the protein that you're working with. So there's an easy way to do that. You need to be able to take a protein sequence like this. So just a random protein sequence. I can count that pretty quickly. That's 9 amino acids in a stop codon. But the genes that you'll work with might be hundreds or longer of amino acids. So I don't want you to do it by hand. So what would you do to figure out how long an amino acid sequence is without doing it by hand? Right, so that's normally what I would do is I would paste it into Word and then use the character count tool to tell me how many characters are in the sequence, and then I'd know how many amino acids it was. If you don't have access to a program that does that, then you could just go to the website that does exactly the same thing. So you paste the protein sequence into carecounter.com, and it tells you how many characters, single letters, single amino acids, are in the sequence. So here finally is the amino acid sequence of the translated nuo1 gene. And what you don't want to do, just for information, when you use TranSeq to produce the translation of a messenger RNA, it will always end with an asterisk. That's representing the stop codon. You don't want to include that in your count of how many amino acids are in your protein, because that's not an amino acid. So when you copy and paste something out of TranSeq, don't also take the asterisk with it. So it turns out this has 479 amino acids in it. Any questions, concerns, comments? What we're going to be doing is taking CDS sequences, translating them, and asking how long they are. Which is sort of boring at first, but I want you to act like Mother Nature. I want you to produce mutations to this gene. And I want you to see what affects those mutations that you define, that you create. How do those affect the amino acid sequence of the protein?